This protocol is optimized to encapsulate pluripotent human embryonic stem cells and differentiate them into dopaminergic neurons in a 3D system. First, treat the colonies with ROCK inhibitor, or RI, and dissociate them into single cells with Accutase. Mix with sodium alginate and pass through the encapsulation device. Continue the RI treatment for 3 days. Transfer the encapsulated cells onto PA6 cells and differentiate for 28 days. Ultimately, gene and protein expression analyses show a higher efficiency of generating dopaminergic neurons.
The major advantage of this particular technique over the existing 2D differentiation protocol is that we try to propagate and differentiate these cells under 3D environment, and that 3D environment provides you an opportunity that cells can be propagated in a high density manner, and then it also gives an opportunity to have a better cell-to-cell interaction for differentiation, and thirdly also give you a better efficiency of differentiation down the track. And also, if I might sort of add that this particular technique can also be adapted for other type of the cells which we can propagate in a 3D environment. Now remember also, like say for example, this particular technique has been optimized based on using a particular ROCK inhibitor that prevent cell apoptosis and also prevent the detachment of the cell from the culture dishes.
Generally, individuals new to this method will struggle in the beginning, as this technique is not typically performed in tissue culture. The following encapsulation technique has been optimized based on Dr. Dean, Dr. Chayosumrit's studies on application of ROCK inhibitor, type of alginate,